You might be wondering, what does dry needling do that might make it work? We must therefore look at the information from physiology research in this area. There are a few findings from previous research that are important to keep in mind while trying to understand the physiology of dry needling. Number one, if you stick a human with a dry needle, this will increase blood flow and blood oxygen saturation to the area. Two, in human patients with chronic pain, there are higher levels of biochemicals associated with pain at myofascial trigger points compared to healthy individuals. Number three, in human subjects with chronic pain, activating a localized twitch response is associated with a spike followed by a net decrease in biochemicals associated with pain and inflammation. Number four, when looking at rabbit models, it is important to note that myofascial trigger points can be identified. Number five, dry needling a rabbit myofascial trigger point with a localized twitch response decreases spontaneous electrical activity compared to dry needling without a twitch response. Let's look closer at a recent study in rabbits. It was well controlled and designed. It compared single dose treatments to 5 treatments over 5 days. It was controlled by a sham group. For half the rabbits, they waited 5 days after treatment to look for changes over time. There was a localized twitch response at the myofascial trigger points. This was an established animal model. In the single dose group, there was an immediate physiological response indicating a decrease in pain sensation, but only a small inflammatory response. 5 days later, these responses disappeared. In the multiple dose treatment group, there was an immediate physiological response with an increase in a variety of biochemical markers for inflammation and stress. There was a decrease in substance P, which, as we all know, is a trigger for pain, and a decrease in beta endorphin, an opioid. This conflicting role between opioid and pain trigger makes things difficult to interpret. At 5 days after treatment, there was an elevation in all markers, including substance P and beta endorphin, which once again makes the interpretation of the data confusing. However, this same animal model was used in a more recent study looking exclusively at substance P as a biochemical marker. This study demonstrated a decrease in substance P 5 days after repeated treatment, which is opposite the results of the previous study. This contradiction has thus far gone unacknowledged by the authors. The major difference between these two studies was the latter study looked exclusively at substance P as a biomarker, thus perhaps changing things by removing confounding variables. Nonetheless, it should be noted that both these rabbit studies demonstrated an immediate short-term decrease in substance P that is consistent with what has been seen in humans with a localized twitch response. But always remember the important limitation: rabbits are not humans. In summary, there is some consistency with single dose but not multiple dose treatments of dry needling. Therefore, there are good early research indications that in subjects with persistent pain, single dose dry needling with a localized twitch response should help decrease pain by altering biochemical markers, but this effect does not last long term. There is still more research needed in order to further evaluate the mechanisms and determine modalities with consistent responses.